Question number first from chapter 3rd is what is replication? So the question was what is replication? Replication. So let's consider this is a DNA, double helical DNA. Now in prokaryotes, there is no nucleus, so this DNA is present in the cytosol, whereas in eukaryotes, let's say a human cell, this DNA is present in the nucleus in the form of chromosome. So we already studied the chapter on cells division, where one cell divides into two. So it is very important that when a cell is dividing, it has to duplicate DNA. So the DNA has to exactly duplicate itself before the cell division, and each daughter cell should get equal number of chromosome or DNA. So let's say if this DNA is duplicating, then this should give rise to two daughter DNA molecules, right? And each daughter DNA molecule should be passed on to each daughter cells. Now this process of DNA duplication is called as replication. Now this replication is carried out by an enzyme called as DNA polymerase. Right? So this is what the question refers to. What is DNA replication? Now I would like to add here that in bacteria, replication occurs in cytosol. Why? Because bacteria has no nucleus, whereas a human cell or a plant cell, right, or a plant cell, which are eukaryotes, they have nucleus. Replication occurs within nucleus. Now, what is the importance of replication? Is of course duplication, but the core value of replication is that the quality of DNA should be maintained from one cell. When it is dividing into two cells, the quality of DNA should be maintained. If it is not maintained, then it can give rise to serious diseases in humans, especially cancers. So so how does replication happens? So let's say hypothetically that this is a DNA molecule, right? Then each one of the DNA molecules, each of the DNA strands, so DNA is made up of two strands, it's a double helical strand. So each of these strands will give rise to, a daughter strand. So fidelity, the loyalty has to be guaranteed, because even if there is a single change in a nucleotide, then it can give rise to serious mutation. So this is essentially the basic of replication. Okay. So what's the next question? The next question is, what is transcription? So here we spoke about replication. So this is one of the functions of DNA. The second function of DNA is that that DNA has to control our cells, the cell functions, cell division. DNA has to control many functions. DNA cannot step outside of the nucleus and do all the functions. So DNA is very critical. Why it is critical? Because it is the molecule of inheritance, and so it has to be maintained. The quality of DNA has to be maintained from one generation, and I'm not talking about one generation, but millions of or hundreds of generations. The quality of chromosome and DNA has to be maintained, and one of the ways it is maintained is that DNA is housed in the nucleus. The chromosomes are present in the nucleus. It cannot go everywhere. But DNA has the message through which it can pass its orders, its message. It can convey through series of steps. And what are those steps? So there are two steps by which DNA can do, and these steps they follow one another. So first is transcription, and transcription is followed by translation. Now, so DNA essentially could start the process of transcription, and then transcription could lead to translation. So what is transcription? So transcription is a process by which the same DNA can work to synthesize a new type of molecule called as RNA. And this, when DNA serves as a template for synthesizing RNA, this process is called as transcription. So this process is called as transcription. Now, RNA is again very sophisticated molecule, very labile. It is very, very delicate. So RNA then again serves as a template for the synthesis of another molecule, which is called as proteins. So I revise. DNA does two functions. One, replication, and the second is transcription, and the end product of transcription is translation. And what does translation means? Translation means synthesis of new proteins. Synthesis of proteins. That's what translation. That's what happens in translation. Synthesis of proteins. Now, what did I mention to you? That in bacteria, the process of replication occurs in cytosol. Why? Because there is no nucleus in bacteria. In humans, the process of replication occurs in nucleus. Same is the case with transcription. So in both the situations, in both bacteria and human nucleus, transcription also occurs in nucleus. Transcription also occurs in nucleus. So I repeat. So in bacteria, replication and transcription both occurs in cytosol. Why? Because there is no nucleus. And in plants and human cells, so let's say in eukaryotes, both replication and transcription, they occur in nucleus. Why? Because the DNA is present in nucleus. So transcription and replication in eukaryotes, both in nucleus. In bacteria, it is in cytosol. Now, this is a very important point. Point is that translation in both bacteria as well as a human cell or a plant cell, translation occurs in cytosol. Why in cytosol? Because the primary site for protein synthesis is ribosomes, and these ribosomes, they are present in cytosol or cytoplasm. So what is the message for this, this whole slide is that DNA is engaged in two processes, two important processes, important in the sense for the survival of the cells, because if there are no proteins, there are no essentially workforce within the cell or a body to do the functions. So cells will not survive. And if the cells want to divide and grow, but there is no replication, again, the system will collapse, the living cell will die. So two important functions of DNA, replication and transcription. Replication is carried out by an enzyme called as DNA polymerase. Transcription is carried out by an enzyme called as RNA polymerase. And so these are the differences between the replication and transcription in prokaryotes and eukaryotes. And translation is a process where RNA serves as a template for the synthesis of proteins. Okay, so let's go to the third question. So third question is, what are the difference between replication and transcription? So I just mentioned to you, I answered this in this just now, that replication is a process where DNA is duplicated by an enzyme, DNA polymerase. And transcription is a process by which DNA serves as a template for the synthesis of RNA by an enzyme. This is carried out by an enzyme called as RNA polymerase. Fourth question is, what is translation? I just mentioned to you just right now. The translation is a process where RNA serves as a template for synthesis of protein. So what's the fifth question? The fifth question is, what are the roles of mRNA, rRNA, and tRNA during translation? So translation is a very sophisticated process. It has to be done meticulously because it involves many things. It involves DNA, it involves ribosomes, and it is an assembly of one of the major macromolecule called as proteins, and proteins are workforce. So this is one of the most important processes that a cell is involved. So, so what cell does? Cell is very smart, and what the cell does is division of labor. So it gives duty to various molecules, and these molecules they do a teamwork, and when they do the teamwork, they lead to the synthesis of proteins. So let me tell you this, why I made this connection is that DNA serves as a most important body of a cell. This DNA serves as a template to make RNA, and that step is called as transcription. And then RNA serves as a template for the synthesis of proteins through process of translation. Now, I just mentioned to you that DNA is always present in a human cells, always in the nucleus. Cytoplasm is always, RNA, or let's say the translational machinery, which is called as ribosome, is always present in the cytosol. So DNA is present in the nucleus, ribosome in the cytoplasm. There has to be some messenger. That messenger is your RNA. But this process of protein synthesis is not simple. It needs more molecule, it needs more partnership. So what did nature do is that nature made 3 kinds of RNA. Not 1, not 2, but nature made 3 kinds of RNA. Now what were those RNAs? Those RNA, one of them was mRNA, right? And as the name suggests, M means messenger. The second kind of RNA was tRNA, and as the name suggests, this was transfer RNA. Transfer RNA, as the name suggests. And the third kind of RNA is rRNA, where R stands for ribosomal RNA. Right? So what happened? I said division of labor, it needs more. So to run a company, a person cannot run company by him or herself. They need a team, they need a collaboration, they need a structure. So cells DNA is very important. So it cannot do all the functions. It needs partnership. It needs more. It needs to employ more people. So what it does, it counts on RNA to have three types of RNA: mRNA, tRNA, and ribosomal RNA. What was the question? How many types of RNAs are produced during transcription? So three types. There are more types, but at this stage, you need to know only three: mRNA, tRNA, and rRNA. So certainly if there are three types, there are three different types, so means they're having three different functions, right? So what are their functions? So this mRNA, it serves as a template for amino acids. Right? What is proteins? Proteins are, you know, from the chapter 1, proteins are macromolecules. And what are the building blocks for proteins? It is amino acids. How many amino acids are there in our cell? At least 20. Right? So 20 amino acids. So how do you know which amino acid, right, has to come to make a protein? That code is present in messenger RNA. So it serves as a template. The second type of RNA is transfer RNA. And the function of transfer RNA is that this transfer RNA brings amino acid to the ribosome. So now you have to think that ribosome is present in cytosol. The translation has to occur on ribosome surface. And what is translation? It is protein synthesis, amino acid. So who will bring the amino acid, right? So the delivery person is tRNA. So mRNA serves as a template. tRNA brings the amino acids. So there is a code that says, okay, this is the sequence of mRNA. So this protein should come. This amino acid should come. Now, if several amino acids came to make a protein, who will make the peptide bond? So the peptide bond between amino acid is made by ribosomal RNA. Peptide bond. Peptide bonds, right? So transcription and translation is a teamwork that is orchestrated by, of course, the starting point is DNA. It is orchestrated by enzyme RNA polymerase that synthesizes RNA. And RNA, they need to do different functions. So there are three types of RNA: mRNA, tRNA, and ribosomal RNA, rRNA. And then these three, they come together to synthesize proteins, and all these happens at the surface of ribosome present on the in the cytosol. So that's why transcription in eukaryote cells happens in the nucleus, but RNA, they have to exit, they have to shuttle from the nucleus into the cytosol and reach out to ribosome on the surface and then get the protein synthesis operational. So let's look at, what's this next question? Yeah, what is the function of ribosomes? So the function of ribosome, as I mentioned, ribosome serves as a platform for the synthesis of proteins. So it serves as a platform, but who are the actors? So ribosomes, it serves as a stage, but who are the actors for protein synthesis? mRNA, that serves as a template. tRNA, that brings amino acid. Then rRNA, that helps in the formation of peptide bonds. Question number 7, which enzyme catalyzes transcription? I mentioned this a couple of times, that the enzyme involved in transcription, what is transcription? DNA to RNA, and the enzyme is RNA polymerase. So RNA synthesis, RNA polymerase. DNA synthesis, which is replication, it is DNA polymerase. So question number 8, pay attention to this question. A DNA molecule has 2 strands, right? Double helical structure. How many strands are used during replication and how many during transcription? So DNA is double helical structure. Yeah. Right? So the double helical structure. In case of replication, each one of the strands, each one of the strands will make new daughter cells, daughter DNA. So each one of the strands will synthesize a daughter strand, right? So this is double helical. Strand number, let's say red strand. Red strand will give rise to purple strand, and then purple will give rise to red strand. Then again, so these 2 strands, so let me tell you again. So what happens that during replication, each one of these strands will separate. So let's say this strand will separate from the purple strand. So there will be a purple strand, they will separate, and then there will be so strands will separate, right? They separated, these 2 double helical strands, they are separate. And then each one of this strand will synthesize a partner for itself. Right? So what is the end result? You get 2 copies of DNA, and each daughter cell receives this duplicated DNA. Now, in case of transcription, what happens that only one strand will make RNA. Here, both the strands, but here, only one strand. Whether it is red or purple, only one strand. So if the purple strand is being used as a template, then if purple, then a red strand will be synthesized. If, let's say, a red strand is being used for synthesizing new RNA molecule, then a purple strand will be synthesized. So the bottom line is, the take-home message is that, so DNA has double helix. In replication, each one of the strand will give rise to a new molecule. So 2 new molecules of DNA are formed. But in case of transcription, only one RNA molecule is formed. If purple is used as a template, then red, and if red is used as a template, then purple strand is formed. So let's look at the next question. What are the difference between transcription and translation occurring in prokaryotes and eukaryotes. So I started my lecture with this differentiation, this difference, highlighting this difference. So transcription in prokaryotes occurs in cytosol. Transcription in eukaryotes happens in nucleus. Translation in prokaryotes is in cytosol. Translation in eukaryotes is also in cytosol. So to summarize, transcription is depending upon presence or absence of nucleus. Translation in both prokaryotes and eukaryotes translation happens in cytosol. Why? Because the ribosome is present in the cytosol. Now the question comes, 10th, what is the relationship between mRNA and genetic code? Now, I mentioned to you that in the earlier slide that mRNA serves as a template for amino acid, right? So let's say if this is an mRNA molecule, and an mRNA could be, just for sake of understanding, let's say AUG, GGG, CCC, UUU. So let's say this is the sequence of ribonucleotides in the mRNA. Now the amino acids that will come for each one of these triple nucleotide is like this. And this is conserved that AUG, in case of AUG, the amino acid will be methionine. In AUG, the amino acid will be always methionine. In GGG, always the amino acid will be glycine. For CCC, the amino acid will be proline. For triple U, it will be phenylalanine. So who does this? From where do we know that AUG for met, triple G for glycine, triple C for proline, and triple U for phenylalanine? How do we know that? We know that because there is a scientific query, investigation, and that scientific investigation revealed that there is a code called as genetic code. And this genetic code decide which triplet nucleotide will make which or code for which amino acid. So let me go to the PowerPoint slides. Can you see this slide? So, yeah, so this is a table called as genetic code table. Here, if you see, I don't want to go into the details. So these are triple nucleotides, UUU. If they come together, it codes for a single amino acid, phenylalanine. For UA, so these are various, this is permutation and combinations. And this table that indicates what triplet nucleotide will give rise to which amino acid. And this table is called as genetic code table. Okay. So the question was, what is the relationship? What is the relationship between mRNA and genetic code? So the relationship is that genetic code tells or reveals the amino acid that correspond to triplet ribonucleotides. So each triplet ribonucleotides on mRNA codes for single amino acid. So that's the relationship. Now here are some examples. So let's see. So these I have given here triplet codes, CAG. So CAG, it makes what amino acid? It codes for rather which amino acid? CAG. So if you go here and look for CAG, so CAG is here. CAG. CAG is here. And this CAG codes for amino acid GLN. GLN stands for amino acid glutamine. Okay. So, and another example on the question was GAG. So GAG stands for amino acid GLU. GLU stands for glutamic acid. So these three nucleotides code for single amino acid. So let's look at the next question. So CAC stands for proline. So these triple nucleotides. Now I would like to pay emphasis on three nucleotides here: UGA, UAA, UAG. Let's say this is the mRNA molecule. This is an mRNA molecule. Now in this mRNA molecule, 90% of the time, 99% of the time, 99.9% of the time, the starting triplet nucleotide will be AUG. So if there is a, so cell is very smart in terms of, for example, you're writing a term paper. What is term paper? If you break down term paper, it is collection of sentences. If you don't use a period or a full stop, then you don't know where, you know the starting point, but we can't make out any meaning. So when you convert your term papers into series of paragraphs, series of subtopics, give it a structure, there's an abstract, conclusion, introduction. So you're giving it a structure. So there's a starting point. Okay, so I know that this is a sentence, and then next sentence, then a paragraph, then the second paragraph. So cells are also very organized. If there is a protein, then there is a starting amino acid, and then there is an ending amino acid. So how does this end happen? So there is a coding flag sequence that says, okay, protein cannot be synthesized beyond this site. So protein synthesis or protein is not made up of unlimited amino acid. There is a restricted number of amino acids within a protein. Okay. So there is a, when a translation is happening, when there is an mRNA strand, and then there is tRNA that brings amino acid from the cytosol, it brings amino acids here. So which amino acid will it bring for AUG? It will bring amino acid methionine. It will bring amino acid methionine. And this methionine will come here. And then let's say a second tRNA comes, it brings amino acid, second amino acid, let's say glycine. So they come together. So all these amino acids, they come are brought by tRNA, and they come here depending upon the sequence of mRNA. Now this has to stop. So even there is a stop signal. So what are those stop signals? So I just mentioned in the question, UAA, UAG, and UGA. All these three nucleotides, three triplet nucleotides, they serve as a stop codon. So a protein, when a translation is occurring on the surface of ribosome, the starting point of any mRNA will be AUG that will code for amino acid methionine. And then after some time, when the protein is synthesized, the process has to stop. Just like, we are driving in a block, we see a red light and we have to stop there. If we don't, then we have to pay the penalty or something else could happen. So it has to stop. This red traffic light or stop codons are three: UAA, UAG, and UGA. So remember, there are three stop codons. So that's what it was, UGA. How many start and how many stop codons are there? So I just mentioned to you that in 99.9% of the cases, the starting codon is AUG. AUG is called as start codon, right? This is only one. There's only one start codon, and that is called as AUG. But there are 3 stop codons. Stop codons are 3 in number: UAA, UAG, and UGA. What's the next question? Yeah. So this is if the DNA strand is written as this AATT. So what will the second strand? So let me copy paste this. I don't think I can paste it on the Google slide, so I'll do it here. Right? So if I'm increasing the font size. So if this is the sequence of DNA, then on one strand, you know, if there is a DNA on one strand, this is the sequence, right? Then if this is the first strand, then question I'm asking you, what will be the sequence of nucleotide on the second strand? So for A, the second strand will have T. For A, T. For T, A. Again A. For G, C. Again C. G. G. C. A. T. A. So if this is the first strand, if this is one of the strand, if this is the sequence of nucleotide on the first strand, the second strand, the sequence will be like this, TTA. Now, same thing, if let's say if the sequence of DNA so the question number 14 is, if the sequence of DNA is AA, if this is the sequence of DNA, then what will be the sequence of RNA? In other words, if this is the sequence of DNA that will make RNA, then what will be the corresponding RNA? The corresponding RNA would be, for A, RNA will have U, A, C, C, G, 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 C, A, C, A, and then T. So there is a relationship, right? We know that A, nucleotide A, it binds with double bond with T. And nucleotide G binds with C by a triple nucleotide, right? So there's a relationship. This relationship is called as complementarity. Okay, so with this we finish the review of chapter 3rd, and so if you have any question, then please ask me. Do you have any question?
Professor.
Yes.
My question is, the way the question was set up, you want us to answer it as a DNA replication, or you just want us to answer as a sample replication? Because you didn't put DNA replication, you just put replication.
So replication is always DNA to DNA.
Okay.
No, there's no other form of replication.
Okay, thank you.